The pursuit of understanding human longevity has entered a new era, one defined by multiomics approaches that enable scientists to explore aging not as an isolated phenomenon within individual organs or pathways, but as a systemic, interconnected biological process. By integrating genomics, epigenomics, transcriptomics, proteomics, and metabolomics, researchers are moving beyond reductionist models to construct comprehensive maps of how the human body ages, adapts, and, in rare cases, resists decline. This systems biology framework holds immense promise not only for identifying markers of aging and health span, but also for discovering actionable targets for intervention. At the foundation lies genomics, which offers a static view of the inherited blueprint. Specific genetic variants, such as those in FOXO3A, APO, SIRT6, and LMNA, are consistently linked to exceptional longevity, suggesting a heritable component to aging trajectories. However, genetics alone explains only a portion of lifespan variation. To capture the dynamic regulation of these genes over time and under environmental influence, epigenomics steps in. By measuring DNA methylation patterns and histone modifications, epigenomics enables estimation of biological age through epigenetic clocks that often predict mortality and functional decline more accurately than chronological age. These clocks are now being refined using panomic data to better reflect the rate of biological wear across tissues. Transcriptomics, which profiles the full range of RNA transcripts in a cell, reveals how genes are expressed in response to age-related cues such as inflammation, oxidative stress, or nutrient sensing. In long-lived individuals, transcriptional profiles often demonstrate preserved expression of stress response genes, efficient DNA repair, and attenuated inflammatory signaling. These patterns suggest not just passive resistance to aging, but active modulation of gene networks that promote cellular homeostasis. Complementing the transcriptome is the proteome, the full suite of proteins that execute cellular functions. Proteomic data reveals which gene products are actually translated and how they are post-translationally modified under stress or aging. Studies show that healthy aging is associated with lower levels of pro-inflammatory cytokines like IL-6, as well as sustained levels of cytoprotective proteins such as heat shock proteins and antioxidant enzymes. Importantly, proteomics can also track the accumulation of dysfunctional proteins that contribute to diseases like Alzheimer's or sarcopenia, offering insights into the interface between aging and pathology. Further downstream, metabolomics captures the small molecule products of cellular processes, amino acids, lipids, sugars, and intermediates of energy metabolism. This layer is particularly sensitive to environmental inputs such as diet, microbiome composition, and physical activity. In centenarians and other long-lived individuals, metabolomic profiles often reflect efficient glucose metabolism, robust lipid homeostasis, and enhanced mitochondrial activity. Moreover, they tend to maintain higher levels of protective metabolites, such as short-chain fatty acids, carnitine derivatives, and NAD+ precursors, all of which are linked to longevity-promoting pathways. By integrating these layers, multiomics approaches illuminate how biological systems interact over time, revealing key nodes of dysregulation or resilience. For instance, linking methylation data, epigenomics, with transcriptional changes, transcriptomics, and protein abundance, proteomics, can expose how aging modifies stress response networks. Metabolomic data can then reflect the functional output of these changes in real time, enabling researchers to identify biomarkers not just of aging, but of successful aging. Systems biology also allows for the identification of distinct aging trajectories within populations. Some individuals may experience accelerated aging due to metabolic dysfunction, while others show declines primarily in immune regulation or cognitive resilience. These patterns, once invisible, are becoming tractable through machine learning applied to high-dimensional omics datasets. Early studies integrating 4 or more omics layers have begun to define aging subtypes with distinct molecular fingerprints and potential therapeutic targets. Critically, multiomics data are also being used to evaluate interventions that aim to slow or reverse aging, including caloric restriction, exercise, metformin, and senolytics. These treatments often produce subtle effects in one molecular domain, but when viewed across multiple omics layers, their systemic impact becomes clear. For example, caloric restriction not only affects energy metabolites but also alters DNA methylation, enhances mitochondrial proteostasis, and reprograms immune gene expression. Multiomics integration helps to validate these outcomes and refine interventions for maximal efficacy. Longitudinal studies are particularly powerful in this context. Projects like the Longitudinal Omics of Aging Project and the Integrative Human Aging Atlas are assembling time-resolved datasets across thousands of individuals to track how molecular signatures evolve before the onset of disease or decline. These efforts are beginning to uncover predictive biomarkers of health span, enabling earlier and more personalized interventions. Despite the promise, challenges remain. Harmonizing data across omics platforms, correcting for tissue specificity, and dealing with interindividual variability are significant technical hurdles. Ethical concerns surrounding data privacy and equitable access to future therapies also warrant careful attention. Nonetheless, as computational tools improve and biobanks grow, the ability to understand and potentially modulate human aging through a multiomics lens is accelerating rapidly. In essence, multiomics approaches mark a paradigm shift from studying aging as a linear, inevitable process to decoding it as a complex, dynamic system open to influence. This systems-level understanding does not just seek to add years to life, but to extend the period of health, independence, and vitality that defines a meaningful old age. By viewing longevity through the prism of integrated biology, we move closer to not only explaining why some people age more gracefully, but also empowering more of us to do the same.